Welcome to Infection and Immunity Evidence Explained, a Doherty Institute podcast. I'm Rebecca Elliott. In a major coup for the Doherty Institute and the Monash Institute of Pharmaceutical Sciences, our two COVID-19 vaccines went head-to-head in a Phase 1 clinical trial in 2022. The interim results have just been published in Lancet's eBioMedicine journal and found that both have strong potential to be an improved approach for boosting immunity to SARS-CoV-2 variants. Here to tell us about these interim results from the trial is Professor Terry Nolan. Terry is a paediatrician and a clinical epidemiologist. He is also head of the Vaccine and Immunisation Research Group at the Doherty Institute and Murdoch Children's Research Institute, which led this Phase 1 clinical trial. Terry, thanks so much for joining me today on Infection and Immunity Evidence Explained.
Pleasure.
Firstly, congratulations on this Phase 1 trial. Tell me about these two vaccine candidates and how they differ to those currently on the market.
Okay, we'll try and be brief. It's different to the existing vaccines, which are now licensed by and large across the world. There are many which are licensed that focus on the whole of the spike protein of the SARS-CoV-2 virus. So these two vaccines are targeted just the tip of that spike protein, which is the bit that attaches to the cells and allows the virus to actually penetrate the cell and do its damage. And that is called the receptor binding domain. And it's much smaller. It's only roughly about a fifth or a sixth of the size of the full spike protein. And it's much more targeted in terms of eliciting an immune response. Secondly, the two vaccines also, because we started this very early. We started actually only a few weeks into the whole COVID disaster in early 2021 and made decisions then as our research in animals moved along about which particular variant to target. Mm-hmm. And at the time, when we had to have the lead time necessary to make these vaccines, the beta variant was the one which was the most worrying. It was the one that really looked like it was escaping protection from existing vaccines from some studies in South Africa at the time, and also was causing potentially more severe disease. Subsequently, of course, we've now seen mutations of the virus go through Delta and then Omicron, and now many of the Omicron subvariants, such as the ones that we're seeing at the moment. But we had the vaccine manufactured against the beta variant. Now, that's also interesting because it has implications for what's called the breadth of protection, the extent to which a vaccine protects against future mutations. So that's what was interesting and unique about what we did. Secondly, when we get to the Phase 1 trial, no one else had done a head-to-head trial before of an mRNA versus a recombinant protein vaccine. And so we were pleased to be able to do that, to actually show both in terms of the safety and also in terms of the immune responses, what they look like comparatively.
Great. And we'll get to that in a little while. But at this stage of the pandemic, obviously there's still a lot of COVID around, but there's a lot of vaccines on the market. Why do we need more at this stage?
We need more vaccines because there isn't enough vaccine to cover the entire global community, even though there are lots of vaccines. It's the way in which they're manufactured, provided, paid for, and so forth. And the extent to which other manufacturers of vaccines, for example, in Africa or in Asia, might want to have access to candidates for the commercial side of development of these vaccines, in a way, we don't have enough. And having more opportunities potentially for safer, more stable or better vaccines is what we should all be looking for, and that's exactly what we've been doing. It remains to be seen, of course, that an academic discovery and evaluation like what we've done with these two vaccines doesn't mean it's a commercial enterprise. In order for these vaccines to reach people, a company or a manufacturer has to pick them up, complete the later phase studies, which are complex and expensive, and then provide manufacture and distribution. So we, as an academic entity, as a research and discovery specialist, and also as an evaluator of vaccines, our job is to actually fuel the pipeline and be sure that companies around the world have opportunities to actually commercialize.
Well, let's take a look at that. I mean, it's absolutely, as you said, no small feat getting a vaccine to clinical trial phase, particularly at speed, and you've done that. What is the process to have got to the Phase 1 trial?
So the animal studies, well, first of all, is the creation of the actual candidates. Secondly, are the animal studies, which are necessary to show that you've got something that works and that there are no safety signals in animal models. By animal, I mean generally small animals like mice and rats. Sometimes in non-human primates. We did not do any studies in non-human primates and other small animals. Thirdly is the extent to which you can then manufacture that vaccine at what's called GMP level. Good manufacturing practice is what that stands for, which means the very, very high standard of purity and quality that's required by the regulators and by various government controls around the world to be sure that whatever you're going to produce is consistent, is stable, and isn't going to introduce any impurities that might endanger health. That manufacturing process is very, very arduous. And in Australia, the opportunity to get that manufacturing done was very challenging. CSIRO made our recombinant protein candidate to this GMP level, and the mRNA vaccine was made by a Swiss company in part, but the formulation of that mRNA was actually done in Melbourne by a Melbourne-based company. So that's all changed now, because I think people will have heard about what's happening with mRNA manufacture in Australia through Moderna and BioNTech and also Sanofi. And that's, in the last year and a half, has revolutionised the Australian situation. So we've caught up very rapidly in Australia. So they were the major challenges. And then to actually mount a phase 1 study, particularly of a product that's brand new and different, is also challenging in terms of the documentation that's required, the very high standards of ethics and safety approvals that are necessary, and all of the things that are associated with that.
Let's just put this into context. So how long would it normally take to get a vaccine to a phase 1 clinical trial?
Obviously, it depends on what the candidate is. If the discovery process to find a candidate, that can take sometimes 10 or 20 years. And it has with, for example, a recent vaccine licensed in the US, about to be licensed in Australia against RSV, respiratory syncytial virus. But if the discovery pathway is fairly rapid, let's say under 5 years, then going from that, doing the necessary later stage animal studies that I've just mentioned, and then into the human study, normally that would be at least 10 years.
Yeah.
And then to go beyond that early phase study into phase 3 studies, through phase 2 to phase 3, you could add another 5 to 10 years to that as well, depending on what the vaccine is and how big the trials are and whether they're problem-free and all those sorts of things. These timelines that people talk about always, if you like, assume that along the way there are no problems. But along the way there are often problems. Sometimes something isn't formulated properly. You have to go back to the drawing board. Sometimes the correct dose hasn't been worked out. Sometimes you need to have other things to add to the vaccine called adjuvants, which might or might not work well, and the formulas for those has to be worked out as well. So it's a complex business, and there is no single predictable pathway. But there's no question that what happened during COVID, including with our own discovery and then implementation of the phase 1, was at the timescale which is unprecedented.
Absolutely. Let's talk about the trial. Firstly, what is a phase 1 trial, and how many phases does a vaccine have to go through before it is available to everyone?
Sure. So a phase 1 study is shorthand really for a first-in-human study. So it means that you have generally a small number of subjects, relatively small number of subjects. It's not based on any statistics in order to achieve what's called statistical power to make comparisons. It's really based on big enough numbers to give a regulator or the discoverers sufficient confidence that they've got something that doesn't have any surprises in terms of the safety. And secondly, that it does more or less do what you intend it to do, which is produce at least a sort of a sufficient immune response. From that, assuming that that hurdle is passed, the next step is to fine-tune the dosing and the scheduling. So for a vaccine, you might need more than 1 dose. You might need 2, or in some cases 3 or even 4 doses of a vaccine. So figuring out that in a phase 2 happens, and also the dose. So phase 2 studies are much bigger, usually small, of thousands generally, but generally they have to be that size to give you enough statistical power to be able to make reliable judgments in choosing a particular dose level over one dose over another, or choosing a schedule, 2 or 3 doses or whatever. And then from that into a phase 3, which is a study that's necessary to measure the impact on the disease that you're actually targeting. So in a vaccine study, you want to prevent something occurring. So to be able to measure the outcome in people who receive control, either a placebo or some other active thing, but just got nothing to do with your target disease, but actually show that the reduction in disease compared to your control is achieved through the vaccine. Those studies, typically in the COVID phase 3s that were done in 2021 by and large, 2021-22 for the licensed vaccines, were between 30 and 50,000 individuals for the phase 3, generally half of whom received the vaccine and half of whom received a placebo. Then there's what's called a phase 4 study, which is where, having been licensed, the vaccines go out into the real world, where there are also very, very large numbers, millions, to allow you to be able to discover if there are any rarer side effects that were not disclosed or found out about in the clinical trials, to find out about the nuances of the vaccine effectiveness, particularly in smaller patient subgroups, such as those who are immune compromised, for example, and so forth, which were in insufficient numbers in the phase 3 clinical trials to reach conclusions about. So that latter thing is what people will recall and understand from COVID. A lot of the data that we had were estimates, particularly from the UK, where they have very, very good data in implementation into the population of how well the vaccines were going once they had been introduced.
It's an extremely rigorous process, and it's obviously got the ethics behind it and everything. And how important is that?
Oh, it's hugely important, and the lay view is that somehow or other there are mad scientists trying to create shortcuts. Nothing could be further from the truth.
Yeah.
That, first of all, the people who are doing this have high integrity, but equally, it's not in their interest to do shortcuts and make mistakes.
Yeah.
The last thing that they want to do is for something to blow back on them down the track because they didn't do their job properly. And all of the, if you like, the custodians along that pathway, the ethics committees, the regulator, the various monitoring groups and so forth within universities, even within the companies, who are also very risk averse. Commercially, don't want to be taking risks either that will lead to litigation or anything coming back against them. So the stereotype of a greedy individual trying to make money here by taking shortcuts is just that, it's a stereotype.
So tell me, who participated in your phase 1 trial and what was involved?
Yeah, so we had 76 individuals in the end who met the eligibility criteria. We had a very strict screening process. So for phase 1s, I didn't mention before, generally you pick people who are in pretty good health. The later phase studies, you may include people who have other medical conditions and so on, which are more like the real world. But in the very beginning, you don't want any other health conditions, if you like, confusing any signal that might emerge about a safety problem with a particular vaccine or a product. So these people who we screened approximately 120 to get to the 76, had to be free of medical conditions. They had blood tests and so on to check on things like liver function tests and so forth. And they're very picky. So even if someone didn't have a disease, but if they had a particular blood level which is just outside of normal range, we would not allow them to take part in the study. And we also, at the time, because we were doing this when COVID was really circulating, we allowed people who had had COVID to be enrolled in our study. The study, by the way, we didn't mention this at the beginning, was a study of these two vaccines as boosters, not as studies in people who had never been vaccinated before. When we started all of this, we had expected, and we started designing everything to imagine that we would have people who are vaccine naive. This was the time when vaccines were not available in Australia, you remember, and there was some concern about their availability and the supply into Australia. But then very quickly, as the vaccines became available, and the uptake was so high, it became apparent that we would not be able to do that study. We wouldn't have enough people who'd be willing to participate who had not been vaccinated. And so as things emerged anyway and the pattern of COVID emerged, it became clear that the vaccines in the future would all be booster vaccines. So in fact, we only gave one dose of our vaccine candidates in our study to the individuals who are randomized to those groups. By the way, they were blinded, so we didn't know and they didn't know what they were receiving. And they received either the mRNA vaccine candidate or the recombinant protein or a placebo. And we also stepped through doses. So we started with a lower dose, made sure it was okay in terms of safety profiles and so on. Then we stepped up to an intermediate dose, did the same, and then we stepped up to a higher dose. That's why it took quite a while to step through all of those different stages.
How long was that process?
It took, in the end, about a year, frankly. And along the way, if people, all lined up to come in, people were allowed to have had COVID beforehand, but they had to have had it at least 3 months before they were enrolled in the study. And so for a number of our individuals, they were all ready to go, had not had COVID, for example, and then 3 days before they were due to be vaccinated, they turn up with COVID. They become ineligible.
Yeah.
So anyway, it was a very tedious process to go through that very carefully. We had what's called a data safety monitoring board. So this was an independent group of people from around the country who looked at our data as it emerged to be absolutely sure there was no safety signal. So we were allowing other people to be the referees to be sure we weren't, if you like, seeing things favourably. and also to give us permission to move to the next step. So that also was another, if you like, staging of this, which actually made it, well, it took longer than we would otherwise normally have done if it wasn't a phase 1. Phase 1, when you're still finding doses and you're not absolutely sure, you go through this very, very carefully.
Yeah. So you mentioned that they obviously could have had COVID, but they've also had received at least 3 doses in prior.
Right. So it was the 4th dose they were getting in this. And to be uniform, we had to insist, therefore, that everyone had the same and that they'd had their last dose, their 3rd dose booster at least 3 months before the study. So otherwise it would have confused the impact of getting a booster, depending on how recently they'd had their own booster of a licensed vaccine.
Yeah. The interim results have just been published in the Lancet eBiomedicine. Yeah. What did you find?
Well, we found, first of all, that both the 2 candidates were safe in terms of the numbers that we studied. So there's a caveat always that I mentioned before. So we're looking here we're not looking for rare diseases because we haven't got enough people or rare potential side effects. But in terms of all of the common things that we were looking at, there was no signal or anything that emerged that was unexpected. The what's called the reactogenicity profile, which means how sore arms were, whether people had headaches, how much discomfort they had for a day or 2 after vaccination, was exactly the same as what we've seen with other licensed vaccines. And then thirdly, in terms of the immune response, what we found was that the boosting was excellent. And it was excellent not just against the target, which is the beta variant, but also against the ancestral strain, the original Wuhan strain. But also we were able to test in the lab against other variants of the Omicron variants. And we also found that the protection against those other newer variants, even though this was a beta-directed vaccine, protection against other Omicron subvariants, including XBB, for example, XBB.1.5 and so on, the more recent ones, was also excellent. So we had our own labs to actually do those, but we also had collaborators at the National University of Singapore who had very sophisticated cross-protection assays, which they were able to bring to bear as well. And they all confirmed that both the vaccines had this breadth of protection, which was very encouraging.
Yeah, that is just fantastic news. So you mentioned in a discussion earlier that the RBD protein vaccine could actually have the potential to be given nasally. Tell me about that.
So the problem with all of the vaccines that we have for SARS-CoV-2 at the moment is that they're injected vaccines. The good thing is we know that they are hugely protective against severe disease. They are not bad at protecting against infection, but they are hopeless at preventing against transmission.
Yeah.
And so people who have the virus and then pass it on to someone else who may be, let's say, vulnerable and so on, more at risk, that's a problem. And that's why the whole issue around isolation and what we went into with all the lockdowns that we had was so important. A vaccine which actually is administered on a mucosal surface, that is into the nose or into the upper airway, produces a response, it also produces a systemic response, which is a protection against infection in the lungs or in the heart or wherever. But what it does, which an injected vaccine doesn't do so well, is induces protection at the site of the mucosa. So that mucosal protection is very important to protect against onward transmission of the virus. And so we know this from other vaccines. So, for example, there's a vaccine licensed now in the US and used widely in Europe and UK as well called FluMist, which is an influenza vaccine. We actually did some of the earliest trials of FluMist here in Melbourne in the early 2000s before it was licensed and in children. And it's a very pleasant little moist, fine aerosol spray into the nostril, which is a lovely way. I've got beautiful pictures of children being immunised. Very different to getting a needle in your arm.
Absolutely. Yeah.
And that vaccine has turned out to be hugely beneficial and successful and is used now, for example, in the school program in the UK, where it's made a huge difference to flu in children in the UK. So we know that those vaccines do work.
We don't have those here in Australia?
No, not yet. Been trying to get it licensed here in Australia. There have been reasons about the company who made that producing a Southern Hemisphere formulation because it was really produced for the Northern Hemisphere market. But when Brazil became a potential customer, Brazil's a huge population also in the Southern Hemisphere, then the case for having it licensed in Australia is building. It's a commercial decision about the company, and I can't speak for them about why it's not yet licensed.
Yeah, of course. Yeah.
So to come back to this recombinant protein, we've got early animal data from our colleagues from our team here, Dr. George Delianis in particular, which show that in mice that there is a very good immune response from mucosal administration, spraying this into the upper airway of mice, that in the test tube, if you like, the immune response looks excellent. Further studies on that are now happening, and recently George has been successful in getting a major grant from the National Health and Medical Research Council to conduct those studies to further evaluate and decide whether we would proceed to human studies of the same recombinant protein but being administered as a nasal spray. We knew this at the very beginning, but we weren't as confident that it would work. We decided to go with the injected form of the vaccine because it gave us the best bang for the buck, if you like, in terms of having a reliable outcome. And in retrospect, probably if we'd had the resources, doing that at the same time might have been a smart thing to do. Anyway, we didn't, but now it's going to happen, and so I'm really pleased that work will continue. There are other opportunities for recombinant protein to be used as well, other than for an intranasal.
Where to next for these vaccine candidates?
Yeah. So for both protein and also the mRNA, they're COVID vaccines. They could also potentially be used in a combination vaccine against other targets such as influenza, for example, or RSV or others. There are other viruses as well. So because we've got a functioning vaccine we know is, from a phase 1 level at least, is safe and also highly immunogenic, produces a very good immune response, it's a very good candidate to consider to be used with someone who has a flu vaccine. So we're hoping that some of the flu vaccine manufacturers around the world will see this as an opportunity if they don't have their own COVID recombinant protein vaccines. Most flu vaccines are recombinant protein or made in a way which produces a protein, including what's called embryo or cell culture. But nonetheless, the protein at the end in combination with another target is a possibility. Now that it's becoming apparent that it looks like we're probably going to have to go into something like an annual revaccination for COVID down the track, it's here to stay, then having a flu and a COVID vaccine in the same syringe, if you like, makes a lot of sense. It doesn't mean it can't work if you have separate vaccines, but in the healthcare sector, providers like simplicity, and if it's something which is a single shot as a combination, it's quite an advantage. We know this from children. We figured out in the late 90s and early 2000s that getting combination vaccines for kids was the best way of improving coverage for families, and that's what's happened over time. So we have combinations now in children. Your children will have received them.
Absolutely.
6, hexavalent, so 6 actual vaccines in one is a standard vaccine that nearly all Australian children receive as young babies. So we know they work, and we know also their development, though, does require further evaluation. You can't just assume that by combining them in the syringe it's going to work. You still have to do human studies. You've still got to show that there's no immune interference. In other words, that the immune response is not in some way compromised by that combination, and also that there is no extra safety factor which hasn't been identified.
And so even taking a step further than that, we've talked about the intranasal route. Could we potentially combine 2 vaccines that way as well?
Yeah, that's an interesting idea. No one's done that yet, to my knowledge. So there you go, Beck.
You've heard it here
There's
first.
An idea. That's right.
There's an idea.
Patent that right now.
Yeah, we'll do.
The other opportunity for the future with the mRNA candidate, which was produced by our colleagues at Monash in MIPS, the Institute for Pharmaceutical Sciences, led by Colin Poughton, is that the mRNA vaccine is not just the mRNA, it's also the lipid nanoparticle. So the lipid component...
What is a lipid nanoparticle for our audience?
A lipid is a fat. Cholesterol is one of the components. Cholesterol is a natural body fat, which we all have, and other similar, if you like, lipids, which have the property of being able to come together and protect mRNA when they're injected. And that's what has made possible. The whole mRNA revolution has been made possible when that discovery was made, because prior to that, it's just not been possible to inject mRNA. It doesn't survive. So when it was figured out that that was how to do it, there was this explosion then of all the things we've seen with mRNA. The chemistry and the science around the lipid nanoparticles and how they're constructed is a whole science in its own. And the opportunity for how they might be deployed in different interventions, including vaccines, but also in targeted cancer therapies and other things, is an area of very active investigation. So Colin has many research partnerships with others in Melbourne at the Peter Mac and elsewhere in the cancer space, for example, in pursuing this. But the science and the uniqueness of what they're offering is also part of their unique contribution. So they have a lipid nanoparticle construct which is similar to but not the same as what's being used elsewhere in the world. And so it too has added value even beyond the mRNA. So both these streams of, if you like, science are things which are now benefited from having been shown in our phase 1 to have been safe. So when it's used with the mRNA itself, that particular lipid nanoparticle also didn't contribute to any safety signal, also enhanced the immune response. So that's another piece of positive evidence to allow that research to proceed as well.
Well, fingers crossed. There's been a huge number of people involved in this project. What's it been like for you?
For all of us, COVID was life-changing. This was certainly. And what it did for those of us working in a university or a research institute environment was made it clear, especially for my basic scientist colleagues. I've been involved in clinical trials for a long time, and if you like, the downstream research. So I was very familiar with this. I've done many clinical trials before, including phase 1s, but I've never done my own phase 1 study of our own product. So learning and doing that ourselves rather than having a huge company to actually provide all of the technical know-how and also the people to actually do the specialist things that were necessary that I described at the beginning of our discussion here today was a very, very big learning curve. We got a lot of help. I was able to hire some outstanding people who had previously worked in industry and who came back into the academic sector who were superb contributors. We used some subcontractors who were commercial and from the private sector to help us with some of the documentation issues and things which are necessary that I mentioned before as well. We had the advantage of having CSIRO to help with the manufacture of the vaccine and the other companies I mentioned for the mRNA vaccine. So for a university to be actually being the conductor of that orchestra doesn't happen very often, frankly. And then to do it under the pressure of time that we're all under and wanting, we didn't know where this was going to end up. We didn't know what the virus was going to do. And of course, we made it happen as quickly as we could. I would have been happy in being able to do it in half the time.
Yeah.
I've always been an impatient individual. And along the way, there were frustrations. Things did take longer than we would have hoped at different times. Australia has got much better at doing things quicker. In the US and in the UK, they probably did things much quicker, helped by having a lot more money, of course. But bureaucracies at all different levels, government and in our institutions, coming to grips with the notion that sometimes you have to do things, not shortcuts, but you have to do things quicker and actually not proceed as per usual practice to actually get to an outcome much quicker. So getting people on board to be able to do that at different times was a challenge. And in the end, it happened. There are lots and lots of different steps here, and getting through them all and keeping that momentum going, dealing with fatigue. All of us were tired. All of us were feeling terrible about the lockdown experience and everything else. But overall, it was an enormously positive experience.
Yeah. All right, Terry. Well, congratulations. All the best for the future of these vaccines, and thanks so much for joining me today.
Pleasure. Thanks very much, Ben.
And thanks to all of you for listening to Infection and Immunity Evidence Explained, a Doherty Institute podcast. This is our last episode for 2023, but we won't be taking a summer break and will therefore be back in your podcast feeds in 2 weeks' time. That's Friday, the 5th of January. For this episode, our first for 2024, I'll be speaking with The Age journalists Aisha Dow and Melissa Cunningham about their new book, Life as We Knew It, which tells the story of Australia's unique experience of the COVID-19 pandemic. If there is a specific topic you would like us to cover in 2024, please get in touch with us by emailing doherty-media@unimelb.edu.au. That's doherty-media@unimelb.edu.au. If you've enjoyed our podcast, please rate and review on your favourite podcasting app and subscribe if you haven't already. The Doherty Institute is a joint venture between the University of Melbourne and the Royal Melbourne Hospital. Wishing you all a very safe and happy festive season. See you next year.